Okay, so Bache, this gets us to our next and an extremely important unit in microbiology, that is systemic bacteriology. This gets us to our next unit, that is systemic bacteriology. देखो systemic bacteriology ना, whenever students study it, दो extreme ends होते हैं. There is one category of students, I guess, like me, who love this topic. I mean, this is one of the most understanding and concept based topics in microbiology. And the second category is the students जो इसको सिर्फ रट्टा मारने की सोचते हैं. Once you just understand this, once you have a hang of this, एक बार मेरे साथ पढ़ रहे हो, you are studying it for the first time with me, you are making your own notes, and then you will be revising those notes, and it will give you immense clarity. एक बार bacteriology में clarity आ जाए, the whole microbiology becomes extremely easy and straightforward. So let us start with our first set of organisms in systemic bacteriology. That is, let us start with gram positive cocci. पहले वाले से शुरू करते हैं. Let us start with gram positive cocci. Let us start with gram positive cocci. देखो, on the basis of catalase test, we are making a flowchart. This flowchart is very, very important. On the basis of catalase test, I can divide gram positive cocci into catalase positive and catalase negative. I can divide the gram positive cocci into two types: catalase positive, and catalase negative. Staphylococcus, staph, that is Staphylococcus is a catalase positive organism versus streptococci, which are catalase negative. So firstly, हमने on the basis of catalase test, we divided gram positive cocci into catalase positive and catalase negative. Staphylococcus, staph, is a catalase positive organism. Catalase test करते कैसे? How is it performed? Catalase test, 3% H2O2 is added to culture colonies. In catalase test, 3% H2O2 is added to culture colonies, and we want to see the bubbles of nascent oxygen. We see the bubbles of nascent oxygen. This is catalase test. 3% H2O2 is added to culture colonies. We add 3% H2O2 to culture colonies, and we look for the bubbles of nascent oxygen. This is catalase. Staphylococci, staph, is catalase positive versus streptococci, which are catalase negative. Next point. Right now we are focusing on staphylococci. Right now we are focusing on staphylococci. So staph, on the basis of coagulase test, I can divide staph into coagulase positive. I can divide staphylococci into coagulase positive, which are the pathogenic staph. Coagulase positive, which are the pathogenic staph, that is, they're responsible for diseases, which are the pathogenic staph, versus coagulase negative. Coagulase negative staph, they are also called as cons. In short, they are called as cons. Coagulase negative staph, or the non-pathogenic staph. Coagulase negative, or non-pathogenic staph. Next, I'm not interested in telling you how a coagulase test is performed. Coagulase test, it is of two types. That is the slide coagulase test, and the tube coagulase test. We are looking at the bound form of coagulase test. यहीं पे छोटा छोटा, let me write it on the side. In coagulase test, it is of two types. It is of two types. Slide coagulase test slide coagulase. Slide coagulase test versus tube coagulase test. Slide versus tube coagulase test. In slide coagulase, we detect bound coagulase. It detects bound coagulase. It detects bound coagulase versus tube coagulase, which detects extracellular free coagulase. Versus tube coagulase test, which detects extracellular free coagulase. कुछ भी नहीं है. Nobody ever will ask you how the coagulase test is performed. This was just for completion's sake and it's a very time pass statement कि coagulase test दो टाइप का है, slide coagulase and tube coagulase. Slide coagulase test, slide पे कर रहे हैं ना, slide coagulase test, it detects bound form of coagulase versus tube coagulase test, which detects free extracellular free coagulase. Not important. What is important is I can divide staph on the basis of coagulase test into coagulase positive and negative. Cons: coagulase negative staph are the non-pathogenic staph. They are the non-pathogenic staph, and these non-pathogenic staph further on the basis of novobiocin test further on the basis of novobiocin test are of two types, that is novobiocin sensitive. They are of two types, that is novobiocin sensitive and novobiocin resistant. Novobiocin sensitive and novobiocin resistant. Staph epidermidis. Staph epidermidis is novobiocin sensitive. Staph epidermidis is novobiocin sensitive versus Staph saprophyticus Staph saprophyticus, which is novobiocin resistant in nature. I am in particular interested about Staph epidermidis. Why?